All right, so let's get this started. This week we're going to be talking about sort of the next step in our sort of dive into how genes are expressed and things like that by looking at genes and proteins, transcription and translation. Now, in terms of things you have to do this week, you obviously have your Labster activity, standard Labster stuff. There's also a survey for exam 1 that I'd like you all to take. At this point, it's not graded, so please just let me know, because I really just want to know what you think, what you thought of the exam. Should I make it easier? Should I make it harder? Well, no one's ever going to tell me to make it harder, but should I make it easier? Do I need to emphasize things better in lecture? Just give me a, I guess a mic check in a sense. Like, just let me know where I can improve to make you better prepared for the second exam and the third exam, obviously. There is an extra credit discussion board due this week, and you obviously have a homework due this week as well. We're about halfway through the semester, which is nice. So we're approaching being done, which I always think is a good thing. So, all right, let's dive in. Genes and proteins. So we're going to be talking today about transcription and translation. So it's going to be fun stuff. Fun stuff. So in terms of genetics as a field, it's relatively old, so you're thinking back to the 1850s or so, but they didn't quite know how to describe a gene, right? They would see these traits, these physical characteristics, but they didn't know quite how to describe things at the genetic level, right? Because we didn't really elucidate the genetics of any organism until the 1950s. So there was like this 100-year period where they were talking about genetics, but they didn't quite know what they were talking about, because we didn't know really what DNA was. And so we'll talk more about this idea of genetics and genes later in the semester when we talk about Gregor Mendel's work. But we're going to start off by talking about the nature of genes. And this really started with Archibald Garrett in 1902. And he recognized that a genetic condition, alkylproteinuria, it's a disease where people lack a specific enzyme. He linked to this idea that it was an inherited disease, and he linked it to this idea that there is a recessive allele. And we'll get back to this idea of a recessive allele very shortly. But this is really where it sort of started in terms of modern genetics, like thinking about genes and DNA and how they relate to overall traits. Now, fast forward about 40 years, we can look at Beadle and Tatum, and they studied a bread mold, Neurospora crassa. So if you've ever had bread spoiled by a mold, it might be Neurospora crassa. There's lots of molds that spoil bread, but Neurospora crassa is a pretty common mold that destroys bread. So what these two scientists did is they used X-rays to damage DNA. Now, as I'm sure you guys all know, X-rays are DNA damaging, right? So when you go to the doctor and say they're worried about you broke your leg or something, right, you get an X-ray. And then you get blasted with the X-rays, right? But the doctor goes, or the technician goes and hides behind a wall, right? It's because X-rays are dangerous, right? And they damage your DNA. So that's what's going on. X-rays to damage the DNA, and they were looking for nutritional mutations. And so what they were doing is they would take the wild type of Neurospora, they would mutagenize it, i.e. blast it with X-rays to try to change the DNA. They would grow it on rich media, and then what they would do is see whether or not it growed on different types of media. And so after they would mutate these things, they would find it wouldn't grow on this type of media, which was called minimal media, which is basically glucose, biotin, which is the vitamin Bs and things like that, and then salts. So it's very, very easy to, very, very simple media. But they found out that, and I will say that, relative to this rich media, which has everything the fungus needs, the minimal media just has the bare minimum to support growth. Just as a note. Now, what they did find is when they supplemented minimal media plus arginine, which is, as you should remember, one of our amino acids, there was growth. So what they were showing here is that by using X-rays to mutate the DNA, they could potentially knock out genetic pathways. And in this case, it would be arginine biosynthesis. So the capacity for this fungus to synthesize arginine. Very important stuff. And so they did a bunch of other stuff as well. So they looked at minimal media plus ornithine plus sertraline, arginosuccinate, and plus arginine. And the idea being is that they thought that you would start with glutamate, and there was a series of genes that had enzymes behind them that would catalyze the reaction of glutamate to ornithine, to sertraline, to arginosuccinate, and then arginine, right? So this is what they were thinking. They were thinking, okay, there's a multiple step pathway in here, and if we mutate this fungus enough, we could potentially knock out specific parts of this pathway. And in this case, what we sort of saw on this one, right? No growth on minimal media. Well, what they were knocking out was a part of this pathway just by mutating it. And so the results from this are, they're pretty cool, right? So you can use X-rays to mutate things, and you can look for physical manifestations of these traits. And that's what they did. And so what this led to was this idea of this one gene, one enzyme hypothesis. And again, when we're talking about genes here, we're thinking about ARGE, right, which produces an enzyme that converts glutamate into, right, or ARGF, which converts ornithine into sertraline or citrulline. Citrulline. I think sertraline is the name of a medicine. I think that's why I get tripped up on that. So that's what they were seeing. One gene, one hypothesis. That the enzymes that were required for the biochemical pathways we just looked at had one gene associated with each one of those enzymes. And remember, enzymes just are proteins that catalyze chemical reactions. And what they were doing by seeing, okay, can I use X-rays to mutate these? Well, I could see how changing one of the genes would change the progression of this overall pathway. So this led to this idea of this one gene, one enzyme hypothesis. And nowadays we think about it more as a one gene, one polypeptide hypothesis, which is really just a fancy word. Polypeptide, remember, is just a fancy word for proteins. That's all it is. So that's where it all started. There's one gene. That gene would be transcribed into some sort of messenger RNA, as we'll discuss, and that messenger RNA would then be translated into an enzyme or a protein. So this leads us to this idea of the central dogma of molecular biology. This was first described by Francis Crick in the 1950s and 60s, and it describes the flow of information in a cell. Now, if you don't remember anything from this course, definitely remember the central dogma of biology. DNA is transcribed via a process of transcription into RNA, and RNA is translated via a process called translation into protein. So it's the idea of taking DNA, which remember is that storage media for all your genetic information, turning DNA itself into a message, and that message into action. And that action is going to be proteins. Now, I will tell you one thing. There are viruses out there. So for instance, HIV actually starts with RNA, goes back to DNA, and then does the central dogma. But everything on the planet uses the central dogma in some way, shape, or form, just as a note. HIV is a pretty important virus, so it bears mention here. But again, this is what we're looking at here, right? For both eukaryotes and prokaryotes, we start with that DNA. That DNA is transcribed into a messenger RNA template. And then that transcription product is then made into a protein via the process of translation. So for eukaryotes, it's moving from the nucleus into the ribosomes. For bacteria, it's moving from the region of the DNA into ribosomes. So that's the flow of information. DNA to RNA to protein. It starts with the DNA, ends in the ribosomes where you make proteins. So let's talk about transcription first. It's a pretty straightforward and simple process, in particular relative to the process of DNA replication. So we spent about 25 minutes last week talking about DNA replication, right? We're going to spend much less time talking about transcription just because it's much, much simpler. But ultimately, transcription itself is what we call DNA-directed synthesis of RNA. And what that means is that we're using the string of DNA here to take a copy of RNA. So and that copy of the DNA, again, is that RNA. So that's the idea. We're directing the construction of mRNA by looking at our original template of DNA. And so when we're talking about transcription, as you all know, DNA is double-stranded, right? And it has directionality, right? 5' to 3'. Now, The idea being here is you can only transcribe one strand of this DNA, and it's typically the sense strand up the top strand, just as a note. But sometimes it can be the bottom strand as well. It just varies between the organism. But that's the idea. We're taking one strand of DNA and transcribing it into a piece of messenger RNA. Now, as you should recall, in RNA, there is no thymine, right? So DNA, as you see, has T, which is thymine. RNA does not have thymine, right? It has uracil. And just as a reminder, remember, DNA and RNA are very, very similar in terms of their structure. They do differ in that sugar, right? Has a deoxyribose sugar, and RNA has a ribose RNA sugar. So they are very similar molecules, but there are some subtle differences to them. But once we physically have that transcription product, then we use that mRNA to directly synthesize our proteins. Now, RNA as a molecule comes in many flavors, but they're ultimately all made from the original starting piece of DNA. So the one we're going to spend the most time talking about today is definitely messenger RNA, and this is the message we make from DNA. Remember. Now, there is special types of RNA called ribosomal RNAs, which are structural components of your ribosomes. We'll talk about those again. We also have transfer RNA, and we'll talk about that very, very shortly. It's kind of a weird thing to talk about without explaining, so we'll get to it in a moment. And then we have these other sort of things, small nuclear RNAs, small recognition particle RNAs, and small RNAs. And we're not really going to discuss these all that much, but I just wanted you to know that they're there. If you want to know more about what they do, definitely keep taking biology courses with me. You'll definitely find out. But for the purposes of today's class, we're definitely going to focus on M, R, and tRNA, respectively. So as you should recall, the genetic code is comprised of A, T, G, and C, or A, U, G, and C in RNA. Now, remember, this was determined by Watson and Crick, right? A always pairs with T, or A always pairs with U, and G always pairs with C, right? That's the sort of the basic building blocks, right, of the DNA pairing. Now, more specifically, if we take a block of 3 pieces of DNA nucleotides, this is what we call a codon. So if I have AAA, that's a codon. Or if I have CCC, that's a codon. Now, the important thing here is that the codons themselves will directly translate to something on the messenger RNA and then directly translate to an amino acid on the protein. So what do I mean like that? Well, let's just start off with a very basic idea, right? So we have sentences with spaces and sentences with no spaces, right? You read this as, why did the red bat eat the fat rat, right? That's pretty straightforward, right? But if I have a sentence with no spaces, right? Right, it's all just one long thing. So there is these breaks, right, in DNA, and they're 3-letter breaks. This is what we call those codons, right? So Y could be AAA, and did could be TTT, right? Now you can imagine the reality of it is if there is spaces, right, you could delete one of the letters and then change the word, right? Why did he, red bat, eat the fat rat? So you can change your DNA as well. So just wanted to point that out. There is essentially this idea of codons, which are triplet pieces of DNA, and each triplet piece of DNA corresponds ultimately to an amino acid by the time it goes up for translation. Now, the other thing we can do to our friends here is we can introduce mutations, and we can do a number of different things here. We can add DNA bases, so we can, if we have AAA, we could throw a T in the middle of that, right? So that we can have an addition. We can have a deletion where we delete something. So if we have TTT, we can delete one of those T's. Now we just have TT. Or we can induce what's called a Frameship Mutation, where we change the way we read the genetic code. And we'll get to that in just one second. But ultimately, you need to remember that the genetic code itself, it's read as A's and T's and G's and C's, but when we're trying to make a product, right, when we're trying to make messenger RNA, when we're trying to make protein, you need to think of it in these triplets, right, these codons overall. So what do I mean by this? Well, let's look at something very, very straightforward, where we're just deleting one base, right? So in this case, we have a messenger RNA strand here at the bottom, AUG, CUA, right, and so on and so forth, right? We read these as triplets, right? AUG in the mRNA has some corresponding thing in the DNA, right? But that AUG, when we try to make a protein from it, corresponds to the amino acid methionine, right? And then CCU corresponds to the amino acid proline, and so on and so forth. Now, if we delete one of those bases, so in this case we delete the G here, so we essentially shift things back slightly. Instead of being met pro ther, we go met pro ser, right? Serine, right? So we're changing completely the amino acids that we're making. Now we could potentially delete 3 complete bases, so get rid of CGC, and instead of having threonine here, we've shifted things back, and now we have met pro his instead of met pro threonine, then histidine. Things, right? If you added 1 base here, you could again change the amino acid, and so that's an important thing. You can directly change your amino acids that you make by making small substitutions at the DNA level. And we'll definitely talk about this more sort of later in the semester, just as a note. So moving on down the line in terms of history, a gentleman named Marshall Nierenberg, and he identified the codons that specify each amino acid. And so as you'll see, and as we've discussed, there are 20 amino acids, and there are 4 DNA and 4 RNA bases, right? And so he discovered how to link each individual 3-letter code to its amino acid. Now, before we talk about that, there are a couple things here. So the first is they're what we call stop codons, and we'll get to this in just a moment in terms of what stopping means. But stopping indicates the end of a protein, basically. And so he identified 3 stop codons, which are UAA, UGA, and UAG. And this terminates the process, again, of translation or protein synthesis. Then we have 1 single start codon, AG methionine. And this always starts the beginning of translation. So this is the beginning of every protein always starts with methionine. So if you remember back from our protein structure, right, the N terminus is always methionine, and the C terminus is always one of these 3. So let's look at this table. And so the way we read these tables, and you probably have seen one of these tables at some point in your life if you've taken any biology courses, but remember our codons are triplets, right? So the first letter is in this left column, the second letter is in the top row, and then the third letter is in the rightmost column. So if I have U, second letter is U, the third letter is U, it's U, right? Or if I have the first letter is U, the second letter is C, and the third letter is C, it's UCC. Right? That's what we're going for here. Now, what you'll notice is that many of the codons have multiple sort of things they encode, right? So UUU encodes phenylalanine, but UUC also encodes phenylalanine. So there is some what we call degeneracy in here, meaning that amino acids are encoded by sometimes multiple triplet codons here. So that's the idea here. And you'll notice some have more, right? So there's only 1 for tryptophan, right? And there's 4 for arginine, right? It just varies. Ultimately, there's 64 total combinations here as you can string together 3 letters, right? 4 times 4 times 4 is 64. Just that's how the math works out. So there's 64 different codon possibilities here, but there's only 20 amino acids. So there is this idea of degeneracy where we have some amino acids are encoded for multiple triplet codons. Now, the other thing I want to mention about this table is that you see it's always U, right? So this is the messenger RNA, right? That means the DNA would be AAA if it was UUU, right? Because remember, the RNA is a direct copy of the DNA, right? And the sort of interesting thing about this genetic code, and I've sort of waxed poetically about how amazing I find DNA as a molecule. But RNA and this genetic code of proteins and their codons is also relatively amazing. And so it is almost essentially universal. So pretty much everything on the planet uses exact same genetic code, which I think is kind of crazy when you think about it, right? The biggest things on the planet to the smallest things, the most complex to the least complex, everything uses that table back there, right? And so you can, just to show you sort of some cute ways you can use this, right? You can use you can use the DNA from a jellyfish to make a pig what we call transgenic, i.e. contains DNA from multiple species, and have it express a protein that makes its nose glow. So you see a wild, this pig that has the jellyfish protein is yellow, and a normal cute pig has the nice pink nose. I will make a mention though, like I know this looks kind of like sci-fi-y. Not this pig is fine. He's not harmed at all. Just as a note, the protein that they use here, green fluorescent protein, is a harmless protein. I just want to put that out there. I wouldn't want the pigs to be hurt either, because I mean, look how cute they are. Anyways, but as I said, pretty much everything uses this. There are some differences. You see them in your mitochondria and as well as in chloroplasts of plants. But by and large, everything uses this everything uses the 20 same amino acids, and they pretty much all use the same exact genetic code. Again, there are some variabilities here, but by and large, most stuff, 99.9% of things use that table. All right, so that's the table. That's the codons. Now let's talk molecular machinery that makes messenger RNA from DNA. And actually, we bring back a friend that should sound familiar. And this friend that should sound familiar is an RNA polymerase. And so it sounds familiar, at least it should sound familiar based upon last class, because as you should remember, the enzyme primase that we discussed last class is also an RNA polymerase. So RNA polymerase should sound a little bit familiar. Now, prokaryotes have a single RNA polymerase, which makes them nice to study, which is fun. And their core polymerase has a couple different parts. So it has two parts that are alpha here. So you see them in sort of these gross mustardy colors here. And then they have the beta, which are these greenish colors. And this is what we call the core enzyme. And then some prokaryotes have this omega, which is here in blue, which is just like this extra stability thing, just as a note. So the idea being is that the whole core of this enzyme is pretty well conserved across all prokaryotes. So it's relatively straightforward and simple. Eukaryotes, I'll just make mention, have oftentimes multiple RNA polymerases. So I think humans have 5 RNA polymerases, if I'm not mistaken. But most prokaryotes just have 1. Now, in terms of the requirements for transcription, we need a couple things. So the first is a promoter, the second is the start site, and the third is the termination site. Now, we'll start with the promoter because this is the initiation point of basically DNA transcription. Or sorry, RNA transcription, sorry. So what the promoter does, it basically acts as like a flag, right? It's like a it's like a giant, like, hey, I'm open, come synthesize me. It's like trying to call people to it. So the promoter itself acts as the slide says, it's a recognition site, and it's where RNA polymerase binds. And so again, the promoter is just trying to recruit RNA polymerase. It's saying like, hey, I'm this gene, come synthesize me. That's basically what a promoter does. And so a promoter itself is found upstream, and I'll show you what that means in just a moment. And the promoter itself is not physically transcribed. So let's look at what I mean by this. So we have the promoter itself. So you see it's a TA, it's a really common type of promoter. It's what we call a TATA box, just as an FYI. I'm not going to test you on it or anything like that, but if you ever see TATA box, it's this. It's referring to this. So you see the RNA polymerase here. It binds to not just one promoter, but a second promoter. So in this case, for this gene we're looking at, there are 2 promoters. One that's at the -10 sequence and one that's at the -35 sequence. Now, this idea of -10 and -35 is a really important thing to remember for the start of transcription, because pretty much all animals and all prokaryotes on the planet start transcription at -10 and the -35 positions. Now, what the heck does that mean? Well, you'll see we have the start site here, right? And so the start site is the start of the gene. This would be the very first DNA or RNA codon in the protein itself, right? So remember, our start codon is methionine, right, UAG. So this would be the very first thing, UAG would start here. Now, what it means to be minus is if this is +1, that means -1 is right next to it. And then there's -2, 3, 4, 5, 6, 7, 8, 9, 10, right? It's just moving back 10 bases from the start site. That's all it is. Now, So that's what we mean by sort of the -10, -35 site for the promoter. But ultimately, as you can see from this diagram, the RNA polymerase binds to the promoters. And the RNA polymerase itself then opens up the double helix at the -10 position. And then once that opens, you have this, what we call that, just to go back to where we were, this omega piece right here, this involved in stabilization. That itself breaks off. And then transcription bubbles have the formation of what's called the transcription bubble, which is ultimately the start site of RNA transcription. I don't know why I'm getting those confused today. It's not a good day for words, apparently. So that's what we're going for. And ultimately, once the RNA polymerase binds to the promoters and it opens things up and that omega unit detaches, transcription begins. So we begin a process called elongation or transcription elongation, which grows your RNA in a 5' to 3' direction. So very much like the way DNA synthesis works, right? 5' to 3'. RNA synthesis works the exact same directionality, 5' to 3'. That's an important thing to remember because we're thinking about this, right? We're working on the bottom strand here, right? Which the bottom strand itself is going to be in a 3' to 5' orientation, and then 5' to 3' is in the opposite direction, right? And so what does this look like? Well, let's dive in. Let's look at this. So we have the RNA polymerase, we have the start site, we have the transcription that's forming. And basically what ends up happening is the RNA polymerase, after it opens up the DNA, it just moves along the DNA. And so remember, when we're building a new piece of mRNA, we're going from 5' to 3'. Right? That's the directionality we're working in. And as you can see, as it opens up, the RNA polymerase just keeps moving. And as it moves, it unwinds and rewinds the DNA into that double helix at the same time, which is pretty cool. But ultimately, it's just making a physical copy of the DNA. And that physical copy itself is in messenger RNA. And remember, they're not different all that much, right? They're simply just differing in very slight ways, right? Just the sugar and just the presence of uracil versus the presence of thymine. And this process of elongation keeps going and going and going, and then eventually recognizes what we call a stop signal. And this tells the polymerase, hey, you're done transcribing this gene, so now you need to physically stop. And so what ends up happening is there's this sort of this hybrid RNA DNA, and again, that hybrid RNA DNA just looks like this, just DNA and RNA bound together. The RNA, essentially, that RNA dislodges from the DNA, the bubble breaks apart, and the RNA polymerase releases the DNA, causing the DNA to reform into that double helix. Now, in terms of termination, we call them terminators, and it's not like I'll be back terminators. They're just like, hey, DNA, stop, places. The simplest terminators are a series of GC base pairs, followed by a series of AT base pairs, but they do vary. Terminators vary relatively widely among prokaryotes and eukaryotes, but again, the most common are GC and AT base pairs. The cool thing about this is they form a hairpin, and the hairpin is just like a bubble in the RNA, which causes that bubble essentially causes the RNA to stop and pause, and then allows the essentially the release of the rRNA. So let's look at what that looks like. So the RNA is doing its thing. Points. Well, again, it's a series of oh my goodness, GCs followed by ATs, and that series of GCs followed by the ATs causes a hairpin to form. And again, this is just a hairpin. Now, once that hairpin forms, it goes, oh crap, I have a hairpin, and then it keeps synthesizing a little bit. It adds 4 more uracils, and then it stops. So it's recognizing the termination point not by like a specific sequence, but the formation of physically of one of these hairpins. Because you can imagine if you're zipping up your a zipper on your jacket, right, and you got like a bubble in the jacket, it would cause you to stop, right? I'm sure you guys have all had broken zipper or something like that, right? As you're zipping up your jacket, right, if you have like a bump that forms or it hops off track, it stops you from zipping up your jacket, right? And that's what's happening here. It's forming a bubble, causing the RNA polymerase to go, whoa, 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 I can't do this anymore. I have to stop. And it causes a termination of RNA transcription. And so that's transcription. Again, very, very simple process. We have initiation, where it binds the promoter, elongation, where we're building the strand, and then termination. And again, termination is just where it recognizes a specific stretch, forms a hairpin, causing the RNA polymerase to fall off. Now, one of the cool things that both prokaryotes and eukaryotes do is they will couple what we just talked about with what we're going to talk about in the next part. And so they couple transcription of RNA to the translation of that messenger RNA into a protein. And so what they do is they form these things called polysomes. So you can see in this nice electron micrograph, as well as this, cartoon version here, where you have a piece of DNA, you have multiple RNA polymerases attached to that piece of DNA, and then attached to each piece of those messenger RNA that's coming off an RNA polymerase, they have physical ribosomes attached to them. So as you should recall from our cellular structures class, what synthesizes proteins is those ribosomes. So we have these polysomes forms, or they're called polyribosomes. And so you can have prokaryotes and eukaryotes synthesizing RNA off one strand of DNA, but translating that messenger RNA into multiple proteins at the same exact time. Now, this isn't super common for eukaryotes, but it is very, very common for prokaryotes. As I'm sure you guys probably know, bacteria respond really fast in the environment. So like if you eat Chipotle that's tainted with E. coli, you'll get sick within 24 hours. Well, it's because E. coli responds really fast. And it can respond really fast because of these polyribosomes or polysomes. So if you ever wonder why food poisoning sets on pretty quickly, part of it's due to the polysomes. So now, in terms of the organization of prokaryotic genes, the prokaryotes themselves are organized into what we call operons. And an operon is just a collection of functionally related genes. So in the beginning of this class, we looked at a pathway to synthesize arginine. That would be an operon, right? We have a bunch of genes that are associated with the synthesis of arginine. And so that's the idea with prokaryotes. They have genes that have similar functions organized into these physical operons. So we'll get more into this later in the semester, but just want to let you know, prokaryotes typically group things together in operons. So multiple genes that help perform a single function. Now, eukaryotes have 3 RNA polymerases. So I overshot my estimate earlier. I thought it was 5, but I guess it's definitely 3. They have 3 RNA polymerases. So the first one, RNA polymerase 1, transcribes ribosomal RNA. RNA polymerase 2 transcribes messenger RNA, as well as some snRNA. And then RNA polymerase 3 transcribes tRNA and other types of small RNAs. And each one of these has its own specific set of promoters. So RNA polymerase 1 only recognizes its own promoter. It can't recognize the promoter of RNA polymerase 2 or 3. So, but let's talk about this RNA polymerase 2, because this is the one that's important for messenger RNA synthesis. So in terms of RNA polymerase 2, the initiation of transcription does require promoters. As I mentioned, for RNA polymerase to be recruited, it needs promoters. Now, for eukaryotes, because we're just extra, because we have to be, we require a series of what we call transcription factors. And now these transcription factors basically help recruit the RNA polymerase 2 to the promoter and physically interact with the gene for expression. And basically what they do is they interact with the gene, they interact with RNA polymerase to form what we call an initiation complex at that physical promoter. So let's just look at what's going on. So we have the promoter, which again, the most common type of promoter is a TA box, right? Just a bunch of T and A's repeating to one another. And we have a transcription factor and other transcription factors coming in. Now, for a eukaryote to physically express that gene that we're looking at here, whatever it could be, we have to have transcription factors bind to the promoter. Now, once those transcription factors physically bind to the promoter, then RNA polymerase 2 can physically then bind to those transcription factors and thus make the messenger RNA from the gene. So a bit different from prokaryotic, right? The prokaryotic one, their single RNA polymerase directly binds to the promoter. But for us and all other eukaryotes, we need transcription factors. We got to make things complicated. Got to make things complicated. Eukaryotes always have to. But as you'll learn, there are benefits to having transcription factors. Not in today's class, but you will eventually learn that there are benefits to having transcription factors. Now, in terms of eukaryotes, they're a little bit different. So they make what's called first a primary transcript. And that primary transcript has to physically be made into a mature messenger RNA. And this is in direct contrast to our prokaryotes. When a prokaryote transcribes a piece of messenger RNA, that is already a mature piece of mRNA. They don't need to modify it at all. It can go directly to a ribosome. So our prokaryotes have an additional step before they make it to the process of translation. Now, the two things that eukaryotes do to their messenger RNA is the addition of a 5' cap. And what this does is it protects from degradation, and it also is involved in the initiation of translation. And they also add what's called a 3' poly A tail. And what this is, it's created by a very specific type of polymerase called poly A polymerase, which also protects from degradation. And so they add these caps, but the other thing they do is actually remove introns or intervening sequences. So I'll show you what introns look like in just one sec, but I just want to show you what these caps and ends look like. So the poly A tail is the easiest to understand. It's just a bunch of A's repeating over and over again. So not much to see there, which I think is nice. But our 5' cap has a little bit more into it. So what ends up happening is the 5' cap is comprised of a methyl group, so a CH3, bound to an amino group here, which is then bound to a 5-carbon sugar. And that 5-carbon sugar is attached to not one, but 2 phosphates, which in turn bind to the phosphate in the very first messenger RNA RNA base here. So this is what the 5' cap physically looks like. Again, very, very, the structure is not that complex, but, much more complex than a poly A tail. But ultimately, again, they're just involved with protection of this messenger RNA. Now, mRNA, it's very, very unstable. Messenger RNA, average cell only lasts about 13 to 15 minutes. And so these caps help add to that longevity. They wouldn't last even 13 minutes if they didn't have these 2 caps, just as a note. So this leads us to the next part of RNA processing, which we would call RNA splicing. So we have to remove pieces of DNA called introns or intervening sequences. And so these are pieces of the DNA, the RNA itself, that are not actually going to be part of the final protein that we make. So they have to be spliced out or removed from our messenger RNA, or what we call our pre-mRNA. Because again, we start with a pre-mRNA in eukaryotes, and then we go to a final product of an mRNA, which then gets translated. So we have our introns, and then we also have our exons. And our exons are actually the pieces of the messenger RNA that we actually want to translate into a protein. And so what this looks like is we have a piece of DNA template. We have exon 1, intron 1, and so on and so forth. That DNA template then is transcribed into messenger RNA. So it is the primary messenger RNA or the pre-mRNA transcript. Now, what you'll notice that we added the 5' cap and the 3' poly A tail, but we still have those introns and exons. Now, introns, again, are intervening sequences. So what ends up happening is they need to be physically cut out of this. And so I hate how they changed the colors between these. I think that was really stupid. I digress. We need to cut out these introns, these purple pieces. So what we do is just physically cut them out. And we splice together all the exons together, which is yellow in here. And so we start with this pre or primary RNA transcript. You add the caps. You get rid of the introns, leaving only exons, which are then tied back together with a 5' cap and a poly A tail. So this is what our mature mRNA for our eukaryotes look like. Remember, prokaryotes don't do this. Actually, I should take a step back. Most prokaryotes don't do this. There are some archaea that do this, but most prokaryotes don't do this. This is mostly a eukaryotic thing in general. And so in terms of how this works, the splicing of pre-mRNAs is accomplished by a piece of protein machinery called the spliceosome. And it does what you exactly think. So what ends up happening, the spliceosome is comprised of short nucleotide RNAs, and they form these base pairs with the 5' ends of our introns. So they basically just bind to them. And what they do is they associate with other factors. So they basically come together to form the spliceosome. And then they form what's called a lariat, which is basically the piece of the intron that they're trying to cut out. And they basically push it out, excising the intron from the physical piece of mRNA, which leads to the tacking together of the exons, thus making a mature messenger RNA. And so that's splicing. And now there's this cool little phenomena in eukaryotes, which is called alternative splicing. And it's not like something like alternative medicine. It's just you can take every messenger RNA product in a eukaryote and splice it slightly differently, getting rid of certain exons and keeping other ones, or vice versa. And so you can essentially alter so just to sort of go back to this picture, I think this one's the best. So if I pretend like I have 1, 2, 3, 4 exons in this picture, well, what I can do is for alternative splicing, what I would do is maybe take 1, 2, 3 and get rid of the 4th, or maybe like 2, 3, 4 and get rid of the first one, right? That's what alternative splicing means. So you can take the same gene, keep different parts of it that you want, and that will create a different function, which is pretty cool. And there are many human disorders that may be due to alternative splicing. So there are a wide variety of them. I'll let you go down that rabbit hole, because there's a lot of genes that are disorders that are associated with alternate splicing. And there has been a study that suggests that 95% of mammalian genes have some form of alternative splicing. So what that means is that if you have 20,000 genes in the human genome, or about that, we do know that in total, just from 20,000 genes, we can make upwards of 93,000 protein products. So alternative splicing adds a whole nother level of genetic diversity to eukaryotes, which I think is kind of amazing, right? Right? Pretty amazing. All right, so moving on, we're going to be looking at the structure of transfer RNA, or tRNA, and the ribosomes. So we're almost to translation. So to synthesize tRNA, we need an enzyme called amino-acetyl tRNA synthase, and we need to do a process called tRNA charging. And so tRNA molecules themselves are, just as their name implies, RNA. But what they do carry on them is not just the amino acid, as it says here, but it also carries what we call the anticodon. And the anticodon is, as its sort of name implies, it is essentially the mirror opposite of the codon. And we'll get into that in just a second. But let's look at a tRNA molecule first. So this is what it looks like as what we call a 2D model. It again is just RNA. And it's just RNA that has formed some RNA secondary structures. And it looks like a T, right? Like a lowercase t. But there's a couple things. There's 3 loops, and we have what's called the acceptor end. And the acceptor end is where we tack on the amino acid, and we'll get to there in just one moment. And then we have this anticodon loop. And remember, this anticodon loop is what corresponds to each individual codon. So if I have a UUU for a codon, there is a matching AAA for the anticodon. That's what that means. Now, 3D model, it looks a little bit like this, less like a T, more like a, I don't know, kind of like an F. Then if you look at a 3D space model, it looks like this. And, we can look at just an icon of it, and it looks like this. So this is what tRNA looks like. Very important thing. Now, in terms of what we do, what's called charging the tRNA, aka attaching an amino acid to the tRNA, we have to use this enzyme called amino-acetyl tRNA synthase. Now try saying that 10 times fast. It's really hard. Now, what it's doing is charging your tRNA. So what we have is we have aminoacetyl tRNA synthase, and we have some sort of, and we have a site for tRNA to land. Now the idea is energy, and we add the amino acid that we're looking for. In this case, we have tryptophan. It has its it is amino group, and it has its carboxyl group, right? That's the conserved structure of all amino acids. So what ends up happening is we use ATP to power this enzyme. So remember, the way this works is we transfer two phosphate groups to anything to add energy to it, right? Because remember, ATP's energy is stored in these first two phosphate groups of ATP. So we use the ATP itself to catalyze the transfer of this amino acid onto the physical aminoacetyl tRNA synthase. That then recruits a tRNA that has not been charged, right? i.e. does not have an amino acid attached to it. Now, what ends up happening is that uncharged tRNA binds to our enzyme, and we use that energy to transfer the amino acid, in this case it's tryptophan, to that tRNA. And then eventually, once that is attached, the tRNA that's been charged, i.e. has its amino acid attached to it, disassociates from this enzyme and goes off to be a part of our protein translation. So the important thing here is that the tRNA synthase, there's one tRNA synthase for every type of amino acid. So there's a synthase for tryptophan, a synthase for histidine. There's one for every amino acid you have, because each amino acid has a very specific binding site here and has a very specific docking site for an uncharged tRNA. And the idea being is you have this anticodon, it has some sort of sequence, and that sequence has to correspond to what the correct amino acid, right? You can't have UUU correspond to tryptophan and, histidine, right? It has to correspond to one physical thing. So that's our tRNA. It has to be charged with amino acid. Just a sort of note, this is where we're charging it, right? We're adding it to this physical end. And again, what we add there has to correspond to the anticodon, which in turn would correspond to the codon. All right, now we've made it to the ribosomes. Now, ribosomes are pretty cool. They have two subunits. They look like a hamburger bun. Now, let's dive into the the ribosome. Let's see what it looks like. We have the large subunit and the small subunit. Now, the cool thing about our physical structure of this ribosomal RNA is it's not just protein. It's a combination of protein plus transcribed ribosomal RNA. So it's RNA that acts as a structural component. So this is what it looks like, large and small subunit. Now, as we dive deeper into what this looks like, there is three distinct sites. We have the E site, the P site, and the E site. Now, these will become much more important in just a little bit as we dive into this process of translation, but just wanted to point it out. We have E site, a P site, and an A site. And if you want to get a jump start on what those mean, they're right here. But we'll talk about them in a moment. Now, the ribosomes' primary functions are twofold. First is to physically decode the messenger RNA. So say if I have UUU, it equals this amino acid. That's what it means by decoding. So I translate this RNA sequence into a sequence of protein, right? The polypeptide chain, right? Amino acids. And then what it also does is it forms peptide bonds. So as you remember, the primary structure of a protein, right? It's just a bunch of amino acids strung together, but those amino acids are strung together by peptide bonds. So what links those peptides together is the ribosome itself. And so the molecular machinery of our ribosome that does that is what we call peptidyl transferase. And this forms the enzymatic together, so forming those physical peptide bonds. Now let's start with our prokaryotes because it is slightly simpler, but relatively similar process as our eukaryotes. Now in prokaryotes, to start translation, i.e. the process of making a protein from our messenger RNA, What we need to do is have a few things. We have an initiator tRNA that's charged with N-formal methionine. So you'll notice methionine is the start codon. So just as a reminder, we need the small ribosomal subunit and an mRNA strand. So this is what we need. So let's look at what's going on. So we have our F-methionine here. We have our mRNA strand, and we have the small subunit. So what ends up happening is we have F-methionine comes in. We have an initiation factor, which is just a protein. It binds to our F-MET, or F-methionine. And then we have an initiation factor that essentially grabs onto the messenger RNA piece. This requires some energy, and so you'll notice we have a new type of energy here. It's not ATP, it's GTP. So it's exactly the same as ATP, but instead of adenosine triphosphate, it's guanine triphosphate. Now we'll talk about guanine triphosphate again in the course when we talk about like signaling and transduction and stuff like that. But just to know, what powers this initial part is physically GDP. And very much like ATP, the high energy and the phosphate bonds is what gives these things their energy. That's what carries energy. Now, once we have the initiation factor bind to the messenger RNA, we have energy to essentially shuttle this along, and it's in the sort of the initial pieces. But we have the start codon AUG, and our messenger RNA binds to that F-methionine, which is UAC, as you can see here. Once F-methionine binds to the start codon, again, which is AUG, this completes what we call the initiation complex. Once that initiation complex is complete, we recruit the large subunit, so the big top of the hamburger bun, that eventually essentially locks in to the initiation complex. And what it does is it puts the initial F-methionine into that P site. And so we have the E site, the P site, and the A site. The very first, the F-methionine that we have is in that physical P site. So that's how we initiate translation. In prokaryotes, of course. Not too different in eukaryotes, but this is how we do it in prokaryotes. So we need some factors, we need some energy, but it's not a particularly difficult process to understand, which I think is good. Now, eukaryotes, it's very similar, as I mentioned, but their initiating amino acid is just methionine. It's not F-methionine, and their initiation complex is much more complex. Just as a note. They have lots more molecular machinery to do it. Like as I said, eukaryotic biology is complex because there's tons and tons of extra proteins and stuff like that, especially relative to prokaryotic biology. So a lot of times we use examples of the prokaryotes just because it's so much easier to understand. Now, once we have the initiation, so again, the formation of this complex, the coming together of the large and small subunits, we have to elongate our protein chain, right? We have to start adding amino acids. So what we do, we have a second charged tRNA. So remember, charged just means it has its amino acid. It then can bind into the A site. And again, just a reminder, the A site is to the P site, right? EPA. So a new amino acid, or a new charged tRNA comes into this A site. So that's the second part. Next up, we need an elongation factor called EFTU, which physically binds to tRNA as well as GTP. And once that happens, a peptide bond can form. And we basically just repeat that over and kind of over again. So what this looks like, we started off, remember, with one methionine in this P site. Well, we have to recruit another one, but the tRNA that we recruit, again, is going to be specific to the RNA sequence here, right? You're not going to just recruit any old garden variety tRNA here. You're going to recruit one that is specific to your RNA sequence, right? So what we end up doing is we have the tRNA, we have a second one next to it. We have to bind those two together. We have to form that peptide bond, and we form that peptide bond from the amino in amino acid 2 to the carboxyl group in the amino acid 1. So what ends up happening is once that bond happens, again, it's enzymatically catalyzed. What it does is it results in the first tRNA, which is in the P site, to be empty or uncharged, and then the second one to have both amino acids tacked onto it. And we basically repeat this process over and over and over and over and over again. And so this is the flow of information. This is how it works. Things move from so we essentially start off with we start off with the initiation, right? And it requires some form of energy, right? And it requires that elongation to keep things going. But once things moved into the P site, then they start interacting with something in the A site, forming a longer peptide chain. And basically the way things go is we started here, they come into the A site, they get the peptide bond to add something else to their chain, then they move to the P site, then another tRNA comes in, that process repeats itself, but what was in the P site then moves to the A site, and once it moves, P site moves to the E site, and once that something moves from E to the E site, then is ejected from the ribosome. And you basically just repeat this process over and over again. Something comes into the A spot, a new peptide bond forms, whatever was in the P spot moves to the E spot, and eventually is ejected from the ribosome. So you basically act as this kind of like crunching thing over and over again. It just moves along the messenger RNA over and over again, just again forming peptide bonds, moving from A to P to E, and then out of the ribosome. So this is what we're looking at here. This is translation, and this is what elongation is. Now, I will mention there is something what we call wobble pairing, or what we call the wobble spot. And the reality of it is most organisms do not carry the full suite of 64 codons, right? So remember, some amino acids are encoded by multiple triplet codons, right? But remember, there's 64 in total, right? 4 times 4 times 4 is 64, right? So there are fewer tRNAs than there are actual physical codons inside a given cell. So what ends up happening, we have this idea of wobble pairing, where there's less stringent base pairing between the 3' base of the codon and the 5' base of the anticodon. And so, oh, I thought I had a picture. I'm so sorry about that. I'll post something to Blackboard. I meant back to add it, but the idea being is that there's this idea of a wobble position. So I'll post something to Blackboard just to make sure that everyone understands this, because I'm sure that explanation was not good enough to do it. And again, I'm sorry. I thought I added a slide here for that. So I'll post something to Blackboard about this wobble position before the end of the day. So once we get to the end of translation, we enter the termination phase, which is basically until termination occurs when the ribosome physically encounters a stop codon. And the stop codons are recognized by what we call release factors, which release the polypeptide chain from the ribosome. So let's look at this. So we have the polypeptide chain. It's growing out the top of the large subunit of the ribosome. So what ends up happening is it recognizes one of those physical stop codons, so UAA and so on and so forth, and it causes not another tRNA to bind, but some of molecular machinery called the release factor. Once that binds, it causes the polypeptide or the newly synthesized protein to break off from whatever is in the P site, and it causes the ribosome itself to essentially fall apart, to thus be then used to make more proteins in the future. So this is how we end. But again, it's all just recognizing one of the 3 stop codons, the binding of a release factor, causing everything to essentially fall apart, which is kind of nice. So that's how we stop translation. So 2 processes, again, they're much simpler than DNA replication, but they are definitely important because they all are tied together in one shape or form here, right? We need to replicate our DNA so that the cells that we make in the future have DNA, but we need to make protein, thus we need to transcribe our RNA, right? So they're all tied together in one shape or form. Now, protein targeting in eukaryotes is something that's unique to eukaryotes because bacteria and archaea don't have organelles. But in eukaryotes, translation, remember, can occur either in the cytoplasm, in the cytoplasmic ribosomes, or on the rough endoplasmic reticulum. Now, there's this really interesting thing that, and we'll go much more into this in a few classes, but There are these sequences at the beginning of a polypeptide sequence, so our protein, that will bind to what we call a signal recognition particle, or an SRP. Now, these SRPs physically recognize, they're physically recognized by the rough endoplasmic reticular receptor proteins. And basically what it does is it holds the ribosome to the physical, the RER. And this begins at sort of that protein pathway we talked about, how things move from the endoplasmic reticulum to the Golgi and then to wherever they need to go. So this is our protein targeting. So let's just look at what exactly do I physically mean by this? So we have the ribosome, it's synthesizing things, right? And it has this signal recognition particle, and the signal recognition particle binds to the peptide. It also sort of partially binds to the ribosome itself, and the signal recognition particle then will bind to the rough endoplasmic reticulum, associating the ribosome with the rough endoplasmic reticulum and allowing the newly created polypeptide to flow into the rough endoplasmic reticulum. The rough endoplasmic reticulum, I should just make, is only rough because you have these SRPs binding ribosomes to it. Otherwise, the ribosomes themselves are just hanging out in the cytoplasm. But they have to physically be bound to an endoplasmic reticulum by these SRPs. And remember, once it gets into the rough endoplasmic reticulum, so the lumen of it, that internal space, then it can then be translated to the transferred to the Golgi, and then from the Golgi it can go anywhere, right? It can go outside of this, it can go to the mitochondria, it can go anywhere. So let's wrap up. Let's summarize gene expression. We have a few, little things to talk about, but let's just summarize gene expression. So remember, it starts with DNA, right? Double-stranded. It has all the genetic information that your cells ever physically could need. Now, to synthesize RNA, we have to use a special enzyme called Rna Polymerase. In eukaryotes, it's Rna Polymerase 2. In bacteria and archaea, it's just Rna Polymerase. Now, remember, we have to make a copy of the DNA. We can't use the DNA to make proteins. We have to make a copy of the DNA, right? We can't risk the DNA getting damaged at all. So we use RNA polymerase to synthesize a primary messenger RNA transcript. Now, in prokaryotes, remember, that primary messenger RNA transcript is comprised only of exons, right? There's no introns in prokaryotes. So once we synthesize the messenger RNA in prokaryotes, it is then directly sent to the realm where it can be translated. However, remember, in our eukaryotes, that RNA molecule that we've made needs two things. First, you need to splice out the introns. Right? You have to cut them out because they're intervening, right? They're not part of the final RNA sequence that we need. And we also need to add that 5' cap and that 3' poly A tail. Once we do that for our eukaryotes, we get our mature or final messenger RNA piece. Once that happens, we send that to the ribosomes, again, which are the site of protein synthesis. Remember, this all starts with initiation factors that cause methionine to bind to the start codon, recruiting the large subunit of the ribosome itself. And the methionine itself, whether it's just regular methionine in eukaryotes or F-methionine in prokaryotes, is always in that middle site, that P. And everything in the ribosome basically moves over the messenger RNA, adding peptide bonds between new amino acids by recruiting new tRNA molecules. And eventually they reach that stop codon. And as they reach that stop codon, it brings in a release factor, which then causes essentially the ending of translation as a process. And remember, that ending of translation as a process means that we have finally formed our protein. So we started with DNA, we made RNA, and we made protein. This is the central dogma of biology, the flow of information in a cell. DNA, RNA, to protein. And this is just a handy table for you that compares your prokaryotes and your eukaryotes. It might be helpful for your homework or your exams or something like that. So I thought it was a nice handy table. So let's talk a little bit more about DNA. Let's talk about mutations before we break off from one another. We've got a couple more slides. We don't have a in-class activity today, so you'll be free with me when we're done with these slides, and I'm sure you guys are all excited about that. Now, mutation as just a process, remember, it's just the change in the DNA sequence. That's it. right? Mutation is not like X-Men, right? It's not like, you know, I got exposed to radioactive material and now I have, you know, claws coming out of my, you know, hands like Wolverine or something. That's not what we're thinking about here, right? Mutations are important, right? They're the driving force of evolution. But again, they're just small and subtle changes in your DNA sequence. Now, there are three classes of mutations. So we have we have our silent mutations where the same, so say we have UUA and that encodes tryptophan. If we change it to UUC, it would still code for tryptophan. So a silent mutation where we'll basically make the same exact amino acid at the end of the day. Now we have a missense mutation, which changes the amino acid inserted. So if we had tryptophan, it changes it to phenylalanine. Now we can do this two different ways. We have transitions and transversions, where we swap a pyrimidine for a pyrimidine, or a purine for a purine. Or we have transversions, swap a pyrimidine for a purine, or vice versa. So that's where we're swapping at the DNA level. But ultimately, it causes a change in the amino acid that is ultimately inserted into the protein sequence. And then finally, we have what are called nonsense mutations, where it's a complete insertion of a stop codon. So instead of having tryptophan, we stick one of the stop codons in there, which itself causes a truncation of protein. So let's just look at what these look like. So we have sort of a regular garden variety thing right here. Right? It's pretty straightforward. Now, a missense mutation, what we're doing is changing, right? We're going from A to T, right? So we're changing things. And it causes, again, a change in the amino acid. So we have arginine at first, now we're changing it to histidine. We have a silent mutation where we swap, you know, the C for the G. And so we have methionine proline, we still have methionine proline. Then we have a nonsense mutation where we go methionine proline to a stop codon as opposed to methionine to proline to tyrosine. Right? So those are our different types of mutations. And you can imagine they have varying levels of effects, right? A silent mutation does almost nothing, right? There's no effects. But a missense mutation might not cause any meaningful changes. And you can imagine, well, maybe if I go from hydrophobic amino acid to hydrophobic amino acid, that's no meaningful change. But you can imagine if I go from hydrophobic amino acid to charged amino acid, that would have a huge impact on my protein. So it just depends on our missense mutations. Now, nonsense mutations themselves, by truncating a protein, can be actually kind of bad. You could have, like, maybe a protein is supposed to be 1,000 amino acids long, and you truncate it after 5. Well, you can imagine that 5 amino acid protein is not going to function particularly well, especially when it's supposed to be something very long. And we can look at just a nice example of this. Well, it's nice because it's, you know, it's easy to understand. It's not a nice disease at all, but many of you might have heard of sickle cell anemia or sickle cell disordered or sickle cell trait, but this is caused by an altered protein structure. So the normal human sequence produces, you know, it's GAG, which produces glutamine, but we have a missense mutation here, where we have GTG instead of GAG. So we're swapping the amino acid from glutamine, which is a polar amino acid, to valine, which is a nonpolar or hydrophobic acid. And so by swapping this out, we completely change the functions. So the normal hemoglobin tetramer looks like this in a normal person, but those that have sickle cell anemia or sickle cell disease, their hemoglobin has this really altered shape. And this altered shape of the hemoglobin causes them to form these long tetramers, which ultimately lead to a sickling formation of their red blood cells. Now, I'm sure you guys probably know a normal red blood cell is circular. For those that have sickle cell, they have these sickle-shaped red blood cells. And again, it's all because of a single point mutation, A to T, causing a shift in the amino acid, causing complete change in the outward expression of this gene, right? Circular red blood cell, clearly different than sickle blood cell. And those that have sickle blood cells, they carry less oxygen. Sickle blood cells get stuck in small capillaries, which leads to numbing and potentially dying off of tissues in capillary that really stuck. So sickle cell, again, really bad disease for those that have it, but again, very simple to understand at the genetic level. A to T causes glutamine to valine. And there are other types of mutations that we'll get into them more as the semester progresses, but there are what we call frameshift mutations, and we've discussed this briefly today. But this is the addition or deletion of the base, which is has typically much more profound consequences because it alters what we call the reading frame. So we talked about this briefly at the beginning of the class, but if I had the cat eats, it would essentially shift to the cat eights, something like that. So it completely changes the way you read a messenger RNA. We can also have what's called a triplet repeat expansion mutation, and this is pretty much basically you're adding a triplet. That's basically what it's looking at, and you basically are adding repeating units to expand the gene overall. An example of this is actually Huntington's disease, and there's many other neurodegenerative disease where you have an expansion of protein, which causes the protein to get overly sticky, causing some pretty severe effects. In addition, we can also look at chromosomal level changes. So we can have chromosomal pieces just be completely deleted. Or, as you see here, we can have duplications where we go BCD and we make an exact copy of BCD. We can have inversions where we change the essentially order, right? So instead of BCD, it's DCB. Now, this might seem trivial, right? This doesn't seem like a big deal, but as you'll learn, the order of genes is very, very important. So if it's supposed to be read BCD and is instead read DCB, that could have some pretty serious issues, right? Because imagine if you're reading a sentence, right, and you just swap the back half of the sentence for the sentence, right? You wouldn't be able to understand that sentence anymore. And that's the idea here. And then the final one, it's not too common, it's called reciprocal translocation, where we're moving pieces of one chromosome to another. This is not too common in most eukaryotes, but you do see this reciprocal translocation in plants quite often, just as a note. And I will make a note that mutations, and I've sort of already made this note as well, but they are involved with evolution, right? They're the starting point. Changes in the DNA lead to changes in traits, right? So evolution is a good thing if it doesn't completely change what you're trying to do. But too much mutation is a bad thing, right? If you completely change or destroy a protein, that could lead to some pretty serious things, right? Something like sickle cell is a pretty serious disease, right? But that's just one mutation. So there is this balance between good mutations and bad mutations, and I don't want to get stuck in your head that mutations are bad. They're definitely not. They are important for evolution. They're important for the functioning of your body, but there's a limit to how good they physically can be. And then the final note is human mutations. Now, human mutation rates, they're interesting. There's a lot of work being done on the evolution of the human genome, and we measure them directly. But the newest sort of work suggests that per human genome, there's 40 to 80 new nucleotide substitution variations per birth. So 40 to 80, point mutations per birth. And insertions and deletions are much rarer. So point mutations are pretty common. And then these large deletions or duplications are very, very rare as well. So point mutations are pretty common. And as we sort of increase the complexity of a mutation, they become less common. And if you want to read some real life examples, I linked this article here. Looking at it talks about what we call human microevolution, how the human population is still evolving, and they talk about in this article about extra arteries. They talk about wisdom teeth. They talk about lactose intolerance. They talk about all sorts of sort of interesting things about human evolution and human mutation. But that brings us to the end of today's class, and I'm sure you guys are all super excited about that. I know I am. I'm always excited when class is over. I'm teaching, but I like being home much, much better. So with that, that's going to be the end of today's class. So if you guys